A very good afternoon to everyone. I hope you must have a very beautiful lunch in this afternoon, but don't regret to have a nap after that, because in this session you are going to learn a lot many things which will help you for cracking the NEET exam or help you to strengthen your concepts. So I am Bushra, your botany faculty, and today I am going to discuss the questions which are mistaken by most of the students in this paper, which is AIATS plus phase 2. Test number 6, which was held on 23rd of February 2025. So in this session, I am basically going to deal with the questions from these topics. First two topics are from class 12. Those are principle of inheritance and variation, microbes and human welfare. So this chapter have very high weightage in the NEET exam, right? Next is ecosystem. This also carry very good weightage. But if we see the next two chapter, which is cell cycle, cell division and respiration, out of these two chapter, from this chapter, almost 4 to 5 question has been asked every year. Same goes with the first chapter. So please be very attentive in this class. Now let's start with the very first question of this paper, or we can say the questions which is mistaken by most of the students in this paper. Now let's start. The very first question, which is 103, which says the cells remain metabolically active but do not proliferate unless they are called on to do so in which of the given phase of the cell cycle. So this question is from the chapter cell cycle and cell division. So now let's see what is cell cycle. So cell cycle is the sequential, orderly, mannered, or we can say the highly genetically controlled process in which cell divide. Cell is the basic fundamental unit of life, right? You all know. Now let's see that what could be the phases of cell cycle. So if we see cell cycle, which is basically divided into two phases. Those two phases are interphase and M phase. Here M stand for mitotic. Further, this interphase is divided into three major phases. Those are G1 phase, or we can say gap 1 phase. Once we proceed in the class, we will get to know that why it is known as gap 1. Next one is S phase, then G2 phase, or we can say the gap 2 phase. Next one is metaphase. Again, it consists of two processes. What are those processes? The first one is karyokinesis. From the name itself, we can see that it is the division of nucleus. Karyon, that means nucleus. Now, next one is cytokinesis. So M phase is basically divided. It consists of two processes, karyokinesis and cytokinesis. Karyokinesis have further phases. They have 4 phases. First one is prophase. Next one is metaphase. Third one is anaphase. All these events takes place in sequence. First prophase, metaphase, anaphase, and the last one is telophase. So the question, if asked that what is the last phase of karyokinesis in M phase, then the answer is telophase, right? If we ask that what is the last phase of M phase, then we could answer that it is cytokinesis. So don't get confused between karyokinesis and cytokinesis, because these are the two processes which are present in the M phase. A very good afternoon, everyone. Right, so this is the basic idea of cell cycle, or we can say the division, that how it proceeds and how complete cell cycle appears like. Now, what are the basic, or we can say what are the major functions that cell do? So if we see that what are the functions that it can do, first it can divide or it will elongate, and or it will differentiate. So सेल के पास सिर्फ 3 ही फंक्शंस हैं करने के लिए. Oops. ये differentiate like this. सेल बड़ा होगा तो डिवाइड होगा, डिवाइड नहीं होगा तो elongate होगा. Once it will get divide and get formed into the specific structure which will be done after differentiation. So all these 3 things can be done. So here we are going to learn that how cell divide. Okay. Now let's start with the interface, that what happens in the interface or the phase in which the most of the things occur. If we talk about interphase and the M phase, and from the complete whole cell cycle, interphase occupy 95% of the complete cycle and M phase has less than 5%. So it is like a drama. If you are going to play a drama, then the preparation is very high. You almost prepare like 3-4 months. So it is interphase, the preparation, and on the day on which you are going to perform is the M phase. So this is how you are going to learn. The actual cell division will take place in the M phase. Let's see about the 95% or the dramatic phase, or in which the preparation is actually taking place. Now let's see. So this is how the diagrammatically representation of cell cycle in this pie form. Here you can see the most of the area from the interface, which is shown here from G1S and G2, is occupied by G1 phase, that is gap 1 phase. In this, if we see that during G1 phase or gap 1, जिसको हम pre-synthetic phase, because it is occurring just before the S phase, उसको ये भी कहते हैं, right? Now let's see कि during this phase what things will happen. During this phase, cells remain metabolically really active. Metabolically active. How? As cell need to divide, so before division all the material which is present in the cell have to get duplicate, right? अगर हमको एक घर को 2 घर में करना है तो जितना भी सामान है we have to duplicate, सबका हमको डबल चाहिए, तभी हम उसको 2 में equally divide कर पाएंगे, right? So during G1 phase those things will take place. All the organelle, or we can say the most of the organelle, sorry, not all the organelle, the most of the organelle will duplicate during G1 phase. Most of the organelle में क्या क्या आ गया? Tell me about the substrates which are used during the replications, during the process of replication. Come on student, comment down that what are the substrates which are required during the process of replication of DNA. Those things will get duplicate during G1 phase. Most of the organelle duplicates. Amino acid synthesis will take place. Obviously energy, etc., all those things. So this is the phase during which all these things will take place. Now the next one is S phase. S phase is synthesis phase. Why it is known as synthesis phase? During which DNA replication or duplication will take place. That means जो हमने DNA replication molecular biology में पढ़ा था वो कौन सा phase में होता है? During the S phase of the cell cycle, right? तो अगर if we consider that during the G1 phase we have this much amount of DNA, then during S phase it will get double like this. Single chromatid will form the 2 chromatids, the duplication of the DNA molecule. Right, along with DNA duplication, centriole duplication will take place during the S phase. G1 में हमने देखा that most of the organelles get duplicate, but out of that centriole duplication, if the question asked, then it will be takes place during the S phase. Right, and the proteins which are required for the binding of DNA, those are also get synthesized during the S phase. Now tell me the name of proteins which help me, which help the DNA for the proper binding. Please comment down so that it could be a really interactive session and we can enjoy this session. हमारी नींद भी जाएगी because this is after lunch session. Okay, so histone protein duplication also takes place during the S phase. From G1 phase, I have asked one question that what types or what substrate get duplicated? It is actually nucleotides. Right, so this is all about the S phase. Now let's move further into the next phase, that is G2 phase, or we can say the gap 2 phase. During this phase, G2 phase or gap 2. So during this phase, the semi-autonomous organelles. Now tell me the names of semi-autonomous organelles which will get duplicate during G2 phase. Along with that, tubulin protein will also duplicate, and the proteins which are required for the further process in the cell division will also take place during this process, during this phase. Right? Along with semi-autonomous organelles, Golgi body will also duplicate. Right? Tell me the name of semi-autonomous organelles which get duplicated during the G2 phase. After that, this is M phase. This region is M phase. As you can see that it is really small area occupying the cell cycle, and it consists of prophase, metaphase, anaphase, telophase, and then cytokinesis, and after again G1 phase will arise. But one thing will happen in this complete cycle, that there are certain cells which do not want to duplicate or divide further, right? So those cells will perform the functions all which will occur in the G1 phase and then exit this G1 phase and go into the metabolically, oops, shit, okay, yeah, I'm back with the complete image, so yeah, right, I thought I mistakenly deleted all the content, but no. So the cells which do not want to proliferate further will exit the G1 phase and goes into the phase which is known as quiescent phase, which will appear here, and that phase is known as G0 phase or quiescent phase. So basically, cell exit the G1 phase to enter this phase. In this phase, only those cells will go जिनको आगे divide नहीं करना है, but they will proliferate जब उनको दोबारा से ज़रूरत होगी. For example, the heart cells and the nerve cells, they do not divide at all, so they are actually present in their G0 phase, right? So this is the case about the G0 phase, which will occur when cell exit the G1 phase and enter into the quiescent phase, during which the cells are metabolically active but they are not proliferating, right? So it is G0 phase. Now let's get back to the question, which is saying the cell remain metabolically active but do not proliferate unless they are called on to do so in which of the given phase of cell cycle. So from the previous idea of the complete cell cycle, I can clearly hit the correct answer, that is G0 phase or the quiescent phase. During G1 phase, The cell is metabolically active but is proliferating because the cell size is actually increasing. As the cell organelle or the number of organelles are increasing, yes Sujit ma'am, DNA for, okay. As in the G1 phase, the size of the cell is increasing, the number of organelles are also getting duplicated. So during this, actually the proliferation is taking place, right? So this is incorrect. S phase, during which the synthesis of DNA or the nucleotides will take place. G2 is the second gap phase where the duplication of the semi-autonomous organelles, those are chloroplast and mitochondria. Along with that, Golgi body also get duplicate. So for the question, rest all options get ruled out and the correct answer is option 1. Now let's see the next question. Okay, so these are the questions which are asked in the previous year in NEET exam. So here you can see that last year only the question has been asked the correct sequence of phases in cell cycle. As I've told you, cell cycle is highly sequential, orderly mannered and genetically controlled cycle, right? That means the sequence will be G1, S, G2 and M phase. So the correct answer is option 1. So these type of questions are also asked in the NEET exam. If you see the next question which says the centriole undergoes duplication during. First one is metaphase. This is the phase from the M phase during the karyokinesis, right? Next one is G2 phase, which is the second gap phase during which tubulin duplication is taking place. Third one is S phase, during which the DNA replication or duplication is taking place. Along with that, centriole is also getting duplicated. So for this question, the correct answer is option 3. Now the next question, which again asked in the previous year only, in the 2024, which says some dividing cell exit the cell cycle and enter the vegetative inactive stage. This is called the quiescent state, that is G0. Just now we have studied in the question given in the slide. And this process occurs at the end of. So this process, the cell is actually exiting the G1 phase or the gap 1 phase from the cell cycle. So the correct answer is option 1. So this is how the question has been asked from this particular topic, from just interphase. So here you have to here you have to just remember that how they are tightly controlled, right? Let's see the next question, which is also asked in the NEET exam in 2023. So here there is match list 1 with list 2. In list 1, the first one is M phase, which is for mitotic phase where the actual cell division is taking place. And it is also known as equational division because in mitosis, what happened? From the parent cell, 2 daughter cells will form and those daughter cells will be identical to the parent cell. That's why it is known as equational division. So here A matches with 4. Next one is G2 phase, which is the second gap phase. Now tell me, during G2 phase, what will happen? Protein will be synthesized. Is it an inactive phase or it is the interval between the mitosis and initiation of DNA replication? Write down in the comment section so that I can know that you are understanding the concept which I had explained just now. कुमारन आप बोल रहे हैं वेरी गुड डायग्रामेटिक एक्सप्लेनेशन प्लस आंसर भी दीजिए साथ में. सो G2 इज द गैप 2 फेज, राइट? तो G2 फेज में क्या हो रहा था? ट्यूबुलिन प्रोटीन सिंथेसिस. सो B मैचेस विद 1. Next one is quiescent stage, which is the another name for the G0 phase, and this is the inactive stage during which cell do not proliferate. So yes, Sukanya Sudarshan, B matches with 1. Last one is G1 phase, which is the gap 1 phase, and it is the interval between the mitosis and initiation of DNA replication. So D matches with 3. So these type of question are also asked in the NEET exam. So you can understand that how important this topic is. But NCERT में कितना छोटा सा दिया है. But please understand the cruciality of this topic. Next one is following are the stages of cell division. So these are the stages which I have told you in the diagram, and we have to choose the correct sequence of stages from the options given below. So the first one is gap 1. Next one is synthesis phase or S phase. Third one is gap 2. Fourth one is karyokinesis और cytokinesis. सबसे पहले न्यूक्लियस डिवाइड होगा या फिर साइटोप्लाज्म डिवाइड हो जाएगा? So karyokinesis will take place and after that cytokinesis will take place. That means division of cytoplasm. The correct sequence is ECADB, right? So this is how we actually hit the correct answer from the questions. Now let's proceed in the next question, which is, yes, Bimal, very good, karyokinesis and then cytokinesis, absolutely correct. Now let's see the next question, which is again from the cell cycle and cell division. Interkinesis is, so we have seen the rough idea of cell cycle initially, right, that how interphase, M phase is further divided. So mitosis takes place in the somatic cell. So if we talk about the formation of gametes, जिसके वजह से we are alive or we are reproducing in such amount. So if we talk about the meiosis, it is divided into two processes. First one is meiosis 1, which is quite complex as compared to the next one, which is meiosis 2. Right, so in meiosis 1, 4 phases are present, again prophase, metaphase, anaphase and telophase, but here as I have written meiosis 1, then their name will be prophase 1, which is having further 5 substages. Then we are having metaphase 1, then anaphase 1, the same with mitosis जो हमने previously देखा, then telophase 1. If we talk about the meiosis 2, again same thing, prophase 2. As I've told you, the process is quite simpler as compared to the meiosis 1. Anaphase. So these are the 4 different phases which will occur during the meiosis 2. If we see the prophase 1, which is the longest phase among all, why? Because it is further divided into 5 substages. Those are, first one is leptotene, the first substage of meiosis 1 from prophase 1. This prophase 1 is really important from exam point of view. Okay, so please try to remember all these steps in the sequence and all the events which are taking place during these phases. Next is zygotene. Third one is pachytene. Fourth substage is diplotene. And the fifth one is diakinesis, the transition phase between metaphase 1 and prophase 1. So these are the 5 substages of prophase 1, and these are really important from exam point of view. So why I have told you all these story? Because in between telophase 1 and prophase 2, after telophase 1 prophase 2 ही आएगा, right? So इनके बीच में there is one process, or we can say one phase exists, which is known as interkinesis. Interkinesis or intrameiotic division. That means within meiosis, there is one phase which is known as intrameiotic division. Right? So these have certain characteristics. Let's see what are the characteristics of this phase, that is interkinesis. So as you can see, it is the phase between the meiosis 1 and meiosis 2, and in meiosis 1 specifically after telophase 1 and before prophase 2. Right? So it is really short-lived phase. During this phase, centriole duplication will take place, but DNA replication will not take place. दोबारा से DNA replication नहीं होगा. Why? Because DNA replication हो चुका during the S phase, which is the part of the interphase. So understand it like that only. So here, no DNA replication will take place. Please remember this point. This is very important. Other tubulin protein synthesis, etc., all these things will take place. DNA, RNA, sorry, not DNA, RNA and protein duplication will take place during the interkinesis, and this phase is again followed by, don't get confused by this word, followed by. That means interkinesis के बाद क्या आता है? It is followed by prophase 2. Right, so these are the feature for interkinesis. Now let's get back to the question, which is actually talking about the interkinesis phase. Now let's see the options. The first one is generally long-lived. Interkinesis, telophase 1 और prophase 2 के बीच में, prophase 2 के बीच में होता है, that means it will be short-lived. तो ये वाला option incorrect हो गया. Next one is the stage between two meiotic divisions. So yes, it is absolutely true. In between meiosis 1 and meiosis 2, this phase exists, and that's why this option is correct. Let's see. Next option, which says followed by prophase 1. As I've told you, don't get confused with this word, followed by. So it is followed by. इसके बाद क्या आता है? It is prophase 1. So this option is incorrect. Next one is the stage in which DNA replication takes place. As I've told you, DNA replication have already done in the S phase. That means during the interkinesis, no DNA replication will take place. Therefore, this option is again incorrect. For the question, rest all options get ruled out, and here we can hit the correct answer, that is option 2. So this is the strategy or the way you can learn the things. Okay. Now let's see the next question, that is 105, again from the same chapter. So from this only you can see that how much weightage this chapter contains from exam point of view. Let's see, the first one, the question says 105, select the correct match with respect to mitosis. Mitosis, I have told you that it consists of two processes. First one is karyokinesis, next is cytokinesis. Karyokinesis is further divided into 4 phases. Let's see what those phases are. I have already told you, let's see in detail. So the first phase of the mitosis, or we can say the M phase, is prophase. G1 हो गया, S हो गया, G2 हो गया, then cell enters into the phase which is known as M phase. So during this, what will happen? There will be condensation of genetic material, right? So condensation of genetic material will start. जितना भी genetic material बना था, वो सब यहाँ पे condense होना start हो जाएगा. Due to which, condensation के लिए क्या चाहिए? Untangling. That means the DNA, which is actually looking like this, completely X-like structure. This is I am making for just make your understanding at ease. So it will get untangled, खुल जाएगा. For example, you have seen the ball of wool like that. It will get untangled and form a ball of wool-like structure, which is. That's why this stage is also known as a spireme stage. Why? Because untangling of complete chromatin will take place and it will appear like ball of wool. Now, during the prophase, the centrosomes, which I have told you, get duplicated during the process of interphase or during the interphase process, will go opposite to the each pole of cell, right? वहाँ पर क्या होगा centrosome का? For example, this is a cell. So in this cell, centrosome is present here towards the at each pole, and they are radiating microtubules like this. दो पोल है centrosome, they are radiating microtubules, and those microtubules get attached with the spindle fibers. This complete structure is known as mitotic apparatus. This complete structure, that means it is having asters. Asters, what do you understand by asters? The centrosome radiating the microtubule. This star-like structure which is made by, in green color, is actually the aster. And these two asters along with the spindle fiber, this spindle fiber will form a mitotic apparatus. Please remember this term and this structure, that how it look like, because it is again very important from exam point of view, right? So this is mitotic apparatus formation takes place during the phase prophase. ये तो बात होगी initial prophase की, that what is happening, condensation is started, that how wool of the ball of wool is formed, due to which it is known as spireme stage, aster formation, centrosome ka duplication, they are moving towards the opposite pole like that. And if we see the late prophase, during which the ER, the nuclear membrane, all those things will get disintegrate. That means by the end of prophase, cell में कोई भी nuclear membrane नहीं present होंगी, all the genetic material will be present all over the cell, right? So ER, nuclear membrane, all those things will get disintegrate. Will get disintegrate. And this disintegration of nuclear membrane will mark the start of next phase, that is metaphase. Let's see here. Okay. Next phase is metaphase. So the complete disintegration of nuclear membrane will mark the start of next phase, which is known as metaphase, during which the complete condensation of chromatic material has been takes place, yani condensation is completed. Condensation of genetic material is completed. That means during this phase, the genetic material is present in highly condensed form. And in this phase only, we can study the morphology of chromosome as well. Question has been asked in the NEET exam again, that during which phase morphology of chromosome can be studied easily. The answer is metaphase. During this phase, one very beautiful event takes place, that is congregation of chromosome. That means all the chromosomes will get condensed and come together on the equatorial plate, imaginary equatorial plate. If we see the cell like this, so here imaginary equatorial plate is present where the chromosome come and lie like this. So if we want to identify the cell, that the cell exists in which phase, if chromosomes are aligned on the equatorial plate like this, then the cell is present in the metaphase. So this is all happen in the metaphase. Now let's proceed to the next phase, that is anaphase. Let me erase all these things so that I can have the space. So anaphase is the third phase of the mitosis, right? And as the chromosomes are actually present on the imaginary equatorial line, then they will start moving towards the opposite pole. How? I will show you here in the diagram. Let's see. If this is a cell, here the chromosomes are present like this. Now they will start, they will dissociate from each other from their point of attachment, that is from centromere. If you remember the morphology of a chromosome, which are attached to each other, chromatids are attached to each other from the point which is known as centromere. So they will get dissociate from each other and move to their respective poles like this. So in anaphase, they will actually look like, this. You can see that centromere is towards the pole and chromatids are trailing behind it. So this is the very important feature for anaphase, as this has been again asked in the NEET exam. So it does actually look like this during the anaphase, which is the third phase. So actual disintegration of chromosome will take place during the third phase of the M phase, that is anaphase. Now, after anaphase, there is the last phase of karyokinesis, that is telophase, during which decondensation of genetic material will take place. That is during telophase. इसके टाइम पे क्या होता है? Decondensation of genetic material. And during this, again, the organelles which get disintegrated during the process, during the event of prophase, will again reform, that is ER, Golgi, nuclear membrane. All these organelles will reform during the process or during telophase. So this is all about karyokinesis in M phase. Now let's see the question. The first one is, as here we have to choose the correct match. The first one is complete condensation of chromosome. Here it is matching them with the late prophase. So as I have told you that during the prophase, as it is divided into two phases, late prophase and early prophase during which the condensation of chromatin material has been started. So it is actually the incorrectly matched pair, because the correct match will be if it is written early prophase. Next one is ER and nucleolus disappeared, early prophase. So this event will take place during the late prophase phase. So again, this is incorrectly matched pair. Next one is decondensation of chromosomes, telophase. So yes, it is absolutely true, as it is the last phase of karyokinesis. Therefore, during which decondensation of chromosomes will take place, where ER, Golgi and other organelle will again reform. Let's see the last one, which says movement of chromatids at opposite pole, metaphase. So this is incorrect, because during the metaphase, which is the second phase of the M phase, congregation of chromosomes will take place and the chromosome appear the most thickest, due to which we can see the morphology of chromosome, and the movement of chromatids at opposite pole is observed during the third phase of the M phase, that is anaphase. So ये option भी गलत हुआ. Therefore, from this we can hit the correct answer, that is option 3. So this is how we proceed to the question. Let's see the next question, which is from the chapter Respiration in Plants, which says how many molecules of ATP are produced through glycolysis by substrate level phosphorylation when one molecule of sucrose enters into the pathway. So what is glycolysis? Glycolysis is the partial breakdown of glucose, right? It is the partial breakdown of glucose, so it is the beautiful diagram from the NCERT itself, the complete glycolytic pathway. So first of all, tell me what is the site of this process, glycolysis? Irrespective of cell, either eukaryotes or prokaryotes, what is the site? What is the site of glycolysis process? So it is cytoplasm. In all the living cell, glycolysis process takes place in cytoplasm of the cell. So it basically consists of 10 steps. As you can see that from glucose to pyruvic acid, it requires 10 different enzymes for facilitating this complete process. Now in the first step, we can see glucose is getting converted into the next substrate, that is glucose 6-phosphate, by utilizing a molecule of ATP, and here a very important enzyme will take place, that is known as hexokinase. Again, question has been asked for this enzyme, that first irreversible reaction of glycolysis is catalyzed by which enzyme? So it is hexokinase, which also requires ATP for the process. Now next, we can see that glucose 6-phosphate is getting converted into fructose 6-phosphate. It is basically the process of isomerization, as fructose is the isomer of glucose. They both have the 6 carbons. So it is catalyzed by isomerase enzyme. Next process is again really important, as this is the rate limiting step of this complete process of glycolysis. Again, this is very important from exam point of view, as this has been asked in the NEET exam. So each and everything is actually written in the NCERT. So read your NCERT like very carefully. Okay. So this process from fructose 6-phosphate to conversion to fructose 1,6-bisphosphate is catalyzed by phosphofructokinase. It is really important enzyme for this process. This enzyme is also known as pacemaker enzyme. Pacemaker समझते हैं? It is put in the human body in replacement of the heart. Basically not in replacement. The functions of heart, which is pumping of blood, is actually done by the pacemaker. So it is actually the pacemaker of this complete process, and it catalyzes the rate limiting step of the process, glycolysis. Now in the next one, we can see that fructose 1,6-bisphosphate is getting converted into 3-carbon molecule, which is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So 6-carbon molecule get dissociate into 3-carbon molecule, and these two molecules are interchangeable. That's why this sign is present. This is DHAP, this is PGAL. Now from this step onwards, the molecules, the ATP molecules will be produced. In the first 5 steps, the ATP, the energy is consumed, right? So this is one important point. Now here the 2 times reaction will take place, one from this and one from here, right? Now let's see that from glyceraldehyde 3-phosphate, it is getting converted into 1,3-bisphosphoglyceric acid by the formation of NADH via NAD positive. So from this, we can see that during the process of glycolysis, 1 molecule of NADH are also produced. From this reaction, if we consider this also, then in total 2 NADH molecules are produced from one cycle, from one process. Now 1,3-bisphosphoglyceric acid is getting converted into 3-phosphoglyceric acid, which is again a 3-carbon molecule, and during this substrate level phosphorylation is taking place. As you can see in the compound itself, यहाँ पे 2 phosphate 3, एक first position पे, एक third position पे. In the next compound, as we can see that it has only 1 phosphate, that is only at the 3rd position. First position वाला phosphate क्या क्या? It is utilized by ATP and helped in the formation of ATP. That's why it is known as substrate level phosphorylation, that means formation of ATP from the substrate metabolite itself. जो भी substrate था उसने ही अपना phosphate donate कर दिया for the formation of ATP. So that's why it is known as substrate level phosphorylation. This is the first substrate level phosphorylation reaction which is taking place during this process. Please remember this reaction also, that while conversion of 1,3-bis phosphoglyceric acid to 3-phosphoglyceric acid, one molecule of ATP is released, as this is the first substrate level phosphorylation reaction of glycolysis. Now next one is 3-phosphoglyceric acid is getting converted into 2-phosphoglycerate. After that, next reaction, there is removal of water. Again, this question has been asked in the exam, that during which reaction the removal of water is observed. So it observed when 2-phosphoglycerate is getting converted into phosphoenol pyruvate. Right, so in the last step of the glycolysis, again the substrate level phosphorylation is taking place, in which phosphoenol pyruvate, or we can say PEP, is getting converted into pyruvic acid. So here also this is substrate level phosphorylation. I am writing SLP for that. So if we see the complete process of glycolysis, so here total 2 substrate level phosphorylation reaction is taking place. First one is here, next one is here. So in total, if we calculate for one glucose molecule, as I've told you that 6 carbon molecule gets split into 2 3-carbon molecules. That means if we say that in the complete process of glycolysis for one molecule of glucose, how many substrate level phosphorylation reaction are taking place, then the answer will be 4. Why? As I've told you that here one substrate level phosphorylation and here is taking place, and here I've told you that it gets split into the 2 3-carbon molecules. When reaction will take place from both the side, then 4 times substrate level phosphorylation is actually taking place. Right? So in the question, as it has been asked for one molecule of sucrose. Sucrose is a disaccharide, that means it is made up of 2 sugar molecules. It is disaccharide. Right, that means glycolysis will take place 2 times, from one glucose molecule for substrate level phosphorylation reaction, for 2 glucose molecule 8 times substrate level phosphorylation reaction will take place. Therefore, for the question, the correct answer is option 2, that is 8. I hope I made you understand that how number 8 came in this question. Any query regarding this question, that how the 8 molecules of ATP are produced through the glycolysis by substrate level phosphorylation reaction? Okay, if you have any doubt, write down in the comment section so that I can clear your any doubt regarding this topic. Okay, then let's move ahead in the next question, which is from principle of inheritance and variation. Okay, so this question is question number 112, and it states that identify the wrong statement for sex determination in honeybee. So sex determination can be of different types in different organisms. So for us, the main type of sex determination type is chromosomal basis of sex determination. Apart from that, 2 other types are also present. One is environmental. There are certain organisms to which environmental condition help in determining their sex. And next one is genic balance theory. These are not at all our point of interest or area of interest. From exam point of view, we are only going to study about the chromosomal basis of sex determination. Sex determination in honeybee is actually different from, actually we can say very different from the humans. How? Let's see. So अगर हम honey bee की बात करें, so in honey bee let's see, one honeybee. Honeybee कितने टाइप की हो सकती है? एक beehive में कितने टाइप के honeybee present होते हैं? So they could be of 3 types. First one is queen. The work of queen is only to reproduce and lay eggs. She is the only one who can reproduce in the beehive. One beehive will have only one queen. Remember that, right? So it is female. Next one is worker bees. These are non-fertile females. They cannot reproduce, but they are female, right? Next one is drones. These are males. They are formed from unfertilized egg. Unfertilized egg. How we will see further. So यहाँ पे जो दोनों females हैं, they both are diploid. That means 2n. If we talk about male, they are haploid. So this is the case. Now let's see that how we can determine the sex in them. And what is the procedure for sex determination in honeybee? If we talk about females and males, as I've told you that they are diploid and they are haploid, right? They have 32 chromosomes, they have 16. Now, via process of meiosis, remember here the process which is taking place for the formation of gamete in the female is meiosis. Meiosis, and if we talk about in males, then from the process of mitosis. From the process of mitosis, it will form the gametes. Okay? So, once the meiosis will take place, which is the reductional division, that means the gamete form will have 2 gametes having 16 chromosomes, like this. As male is dividing its gamete via the process of mitosis, which is the equational division, that means the gamete will have 16 chromosome only. So if this egg remain unfertilized, or we can say by the process of parthenogenesis, by the process of parthenogenesis, that means no fusion of male or female gametes, then there will be formation of drones, or we can say males, which are haploid, right? And if there is process of fusion, or we can say the fertilization will take place, then there will be production of female worker bees, which are diploid in nature. These are diploid in nature and they are result of fertilization. This is really important again from exam point of view, right? So remember that female here will reproduce the gamete by the process of meiosis, where it will produce the gametes having half the number of chromosomes as present in the parent body. And if we talk about the male, then it will produce the gamete by the process of mitosis. And when egg remain unfertilized, and by the process, or we can say by the means of parthenogenesis, it will form the drone. Then males are formed, which are haploid. The roadster is saying, ma'am, please explain Drosophila cross after this. Yeah, of course. Now, where I am, so yes, unfertilized egg will lead to the formation of drones, which are sterile and they are haploid. So like this way, it is the sex determination in honeybee, due to which the drones do not have father, neither they have son. please remember this line really carefully, that zones which are formed, which are haploid in nature and they are formed by the means of parthenogenesis, they do not have father. They do not have father, neither they have son. ना ही फादर इनके पास होता है, ना ही सन होता है. But what they have? They have grandson and grandfather. Due to this type of sex determination, they have grandson and grandfather, but उन पे father और son नहीं होते. This statement has been asked in the NEET exam previously. So please remember that zones which are haploid in nature do not have father, neither have son, but they have grandson and grandfather. If you proceed further, that how that they are not formed by the process of fertilization, that's why they don't are having father. They are only having mother, right? So like this way, and here there is one beautiful wasp which is roaming here. Now let's see the question, which says the wrong statement for sex determination in honeybee. Let's see the option. The first one is females are diploid. So yes, it is absolutely correct. Next one is both male and female bees show meiosis to form gametes. As I've told you, meiosis will be shown by the females, but males will show mitosis for the formation of gametes. So this option is incorrect. Next one is males do not have father. Yes, as I've told you that males do not have father, neither they have son, but they have grandfather and grandson. Next one is male develops through parthenogenesis. Again, this is true because they are formed from the unfertilized egg, directly from the egg formed by the queen bee. Right, as the question is asking about the wrong statement, therefore for the question we can easily hit the correct answer, that is option number 2. Right, so that's all for this class. I hope I made you understand all the concepts from the cell cycle and cell division and from the principle of inheritance and variation. Please stay hydrated and stay tuned for the next session. Thank you so much for watching this video.